Hi students, today we will see the composition of blood. So for clinical biochemistry, blood is a very important component. We should know first the composition of blood to study all the other diseases regarding infections or changes in blood. So blood is the fluid connective tissue. Blood is the fluid connective tissue, an important channel through which all the nutrients, waste materials reach their respective target materials. So let's see the physical properties of blood. This you can write on your own: color, volume, pH, specific gravity and viscosity. These are just as an introduction. You can give physical properties of blood. The color differs between arterial and venous blood. Arterial blood is scarlet red and venous blood is purple red. And volume up to 5 liters. pH 7.4, slightly alkaline. Specific gravity 1.060. Viscosity some 2 to 3, but 5 times more viscous than water. That is a specialty. These are all the 5 physical properties of blood. Blood is a fluid connective tissue. This is the most important point. Okay. So next, coming to the functions of blood, because before going to the actual composition, the functions of blood, regulation of, the most important function being the transport. The blood transport the nutrients, that is absorption. Studies have seen absorption of all the amino acids. The end products are amino acids, glucose and glycerol and fatty acids. All the end products of digestion are distributed to various tissues through blood only. And the waste materials, of course the gases or the waste metabolic end products, they enter into the blood, and through blood only they reach the either kidneys or the lungs. Lungs means CO2 will be exhaled, and the other uric acid and other waste materials, they reach the kidneys and they are eliminated. So that is also a transport function. And different hormones are secreted by the endocrine glands and released into the blood, and they reach their target tissues through blood only. So the blood is the main channel, transport channel through which all the nutrients, waste products, hormones reach their specific target organs. And exchange of gases is another important function of blood. As you know, transport of O2 and CO2 between lungs and peripheral tissues. And another function, fluid balance and pH balance. pH balance means acid-base balance. The blood maintains different acids and bases are being released into the blood, but different buffer systems are present in blood which maintain the pH of the blood. Fluid balance, of course, this is another heavy topic. Fluid balance means the fluid is regulated between, or maintenance of regulation between blood and tissue spaces. If at all there is any disturbance in this fluid balance, we get inflammation, okay, edema, that is retention of water in tissue spaces, if there is a dysfunctioning of a fluid balance by blood. So that is called Starling's hypothesis. So blood also maintains fluid balance between blood and tissue spaces, pH balance, acid-base balance due to various buffer systems. And next, regulation of body temperature. Okay, so through circulation, because the blood flows continuously in blood vessels, there is a body temperature regulation. The temperature gets distributed between the tissues. There is no high temperature one place and low temperature one place. Uniform temperature throughout the body due to circulation. So next, defense, that is the blood contains antibodies or immunoglobulins which provide the defense mechanism or the protection of the body against infections. So next, prevention of hemorrhage. This is the self-defense mechanism. Blood contains different clotting factors which prevent their own loss. Okay, so blood lone clotting factors untai kabatti tana loss ni tane prevent cheskuntundi. That is prevention of hemorrhage. That is blood clotting. We will see the next topic. So through blood clotting, because the blood contains internal blood clotting factors, it prevents its own loss. That's all prevention of hemorrhage. These are all functions of which transport and exchange are very important functions. Okay. Now coming to actual composition of blood. Blood contains 55 to 60% the watery content, which is called the plasma, the straw colored fluid, and the cellular components, which are also called the formed elements of blood, which contain RBC, WBC and platelets. Okay, they comprise only up to 40 to 45%. They are the cellular components of the solid components. This is the liquid portion of blood, which is called plasma. And plasma in turn contains 92% water and only 9% solids. And these 9% solids have enormous components, of which inorganic and organic. Apart from that, I have written some elements here. So much of the components of the blood are mostly present in the plasma. So the inorganic components, most important cations, sodium, potassium, calcium, magnesium and anions, chloride, bicarbonate ions, phosphate, etc. Gases, oxygen, O2, CO2 and iron, copper, phosphates. Okay, you can write down. I am dictating even though you can't see here. So the composition question is itself is composition. So I have to write number of components of blood. So the organic components include the proteins which include albumins, globulins, fibrinogen, most important proteins which are also called plasma proteins. And the most important carbohydrate in plasma is glucose, as all of you know, fats, okay, triglycerides, phospholipids, cholesterol. They are also present in plasma. NPN, non-protein nitrogenous substances. Okay, other than protein, nitrogen is present also in the form of certain substances. Urea, creatinine, uric acid, ammonia, thyroxine, epinephrine, bilirubin, creatine, urea and uric acid. Most important all these three. And blood NPN antamu, non-protein nitrogenous substances. All these are present in plasma. They comprise up to 9%. Actually, the blood comprises 8% of our total body weight. 8%. Volume, I think, 5 to 6 liters. Okay. So, the various inorganic and organic components. And these cellular components are called the formed elements of blood. And other than organic and, of course, organic lo kostayi ivanni, hormones, enzymes. Enzymes are carbonic anhydrase, lipase, esterase, protease, amylase. All these are present in plasma. Hormones, as you know, all the hormones circulate through blood. Antibodies, immunoglobulins, they also come under plasma proteins. Antibodies are also present in the blood. Acids, these are all metabolic products, pyruvate, lactate, phthalate, citrate, beta-hydroxybutyric acid, okay, glutaric acid. All the acids, different acids are also present in plasma. Blood is a vast tissue which comprises so many elements of the body. Next, we can go to red blood cell. So red blood cell in detail. So these are the major components of blood. They are also called erythrocytes. They are present up to 4.5 to 6 million per cubic mm. Okay, largest or most abundant cellular components of blood. All of you know. So as the name indicates, it is a red in color, red blood cell. The most important property is that it doesn't have nucleus. Non-nucleated shape is biconcave discs. The shape is somewhat like this, and with an average thickness of 7.5 microns. Okay, nucleated, non-nucleated, biconcave in shape. Okay, that shape is very facilitative for them to squeeze through the narrow capillaries. Okay, flexible ga undalante aa shape most convenient shape. It has, nature has selected such a shape, and the gases O2 and CO2 transport is also very facilitative for such shape. Okay. Next, 100 to lifespan is 100 to 120 days. These are all information. As far as composition of blood, you have to remember so many points. Lengthy answer, good marks. Okay, the most important protein present in the blood which transports, which helps for its function of transport of oxygen and CO2 is hemoglobin. The most important protein, and the remaining is made up of carbohydrate, small amount of proteins, enzymes, which is called the stroma network, RBC stroma. Okay. And its osmotic pressure is equal to the osmotic pressure of plasma, which is equal to saline, that is 0.9% sodium chloride. Okay, the pressure exerted by this solution is called saline. This is actually the saline. The pressure exerted by this solution is equal to the OP of RBC, which is also equal to plasma. The plasma and RBC osmotic pressure should be the same. Okay. And the formation, the origin, development and maturation of RBC is called erythropoiesis. I said origin, development and maturation of RBC is called erythropoiesis. And this takes place in the bone marrow from the stem cells, and stem cells give rise to all types of the formed elements or the cellular components of blood. And the factors necessary for erythropoiesis are erythropoietin, hormone secreted by kidneys, and vitamin B12, okay, cyanocobalamin and folic acid. These three are the important necessary factors for erythropoiesis or formation of RBC. It takes 7 days for formation of RBC. Okay, lifespan is 100 to 120 days. Most important function being transport of oxygen and CO2 across lungs and tissues. And increase of RBC, increase in number of RBC. Variations in RBC occur in certain diseases. Increase is called polycythemia. Decrease of RBC, you know very well, is called anemia. Okay, when your blood content decreases, your RBC number decreases, that creates a situation called anemic anemia. Okay, next, other than these components, the main important components of RBC are glutathione, which maintains the structure of RBC or RBC membrane. RBC membrane integrity kaapadedi glutathione. Okay, methionine, creatine, urea, glucose, amino acids, enzymes of glycolysis and HMP shunt pathway. Ivanni kuda RBC stroma lo untayi. So, see, enzymes, the RBC derives its energy for function from these two pathways, glycolysis and HMP shunt pathway. Okay, and next, RBC membrane has a specificity. It contains proteins like spectrin, ankyrin and glycophorin. Spectrin, ankyrin and glycophorin. They have a very special property in maintaining the structure of RBC. Okay, these are the RBC membrane proteins. And next, the properties of RBC are Rolox formation. Rolox means stacking up, piling up of one above the other. This is the actual diagram. You keep coins one above the other, that is called the Rolox formation. When the blood is drawn outside and kept in a test tube, immediately all the RBC form a Rolox-like structure. That is the property of RBC, Rolox formation. Crenation, that is shrinking of RBC due to surrounding conditions, differences in the osmotic pressure of the conditions. Shriveling or shrinking of RBC due to evaporation of the plasma. And hemolysis, this is breaking up of RBC and letting the hemoglobin out. This occurs due to certain drugs and certain poisons also. Hemolysis. And agglutination, as you know, you get in immunology also, the clumping of RBC and when they encounter certain antigens, that agglutination occurs due to the antigen antibody complex. These four are the properties of RBC. Okay, so the most important function of RBC is the transport of gases between the lungs and the tissues. Okay, so their membrane is very much flexible. RBC membrane and shape is very much flexible to carry out that function. So now, leukocytes, the second component of blood, leukocytes. And here, leukocytes are the WBC, white blood cells. Mostly, they help in the defense or the defense mechanism of the body. And RBC, as you know, just we discussed only transport of O2 and CO2, nothing to do about defense. These are all defense mechanisms which our blood possesses. So leukocytes, based on the presence or absence of granules, it is divided into granulocytes and agranulocytes. Presence or absence of granules. They are called polymorphonuclear leukocytes, mononuclear leukocytes. The name itself indicates polymorphonuclears. Nucleus is bilobed or multilobed. Okay. They are again divided into neutrophils, eosinophils and basophils. Agranulocytes or mononuclear leukocytes are divided into monocytes and lymphocytes. Okay. So the neutrophils, their lifespan is 2 to 5 days and they are concerned as the first line of defense in the body by the action of phagocytosis. They engulf the foreign particles and destroy, and they make the first line of defense mechanism of the body. Neutrophils and monocytes, they form the first line of defense in the body. They protect us from infections. And eosinophils, their lifespan is up to 7 to 12 days. They act against parasites by detoxification and disintegration of the foreign particles. Okay, and regarding basophils, the lifespan is 12 to 15 days. They are mostly found in the wound healing processes after any inflammation like that. Okay, so these are the various diagrams. You can see clearly the granules, and you should describe about the nucleus also. Okay, here multi-lobed nucleus, here and here bilobed nucleus. Okay, at all the functions here. Okay, you can see the granules. Basophils, because they are when we treat them with dyes, like basic dyes like methylene blue. Okay, they give the color. And eosinophils, they stained red or orange when stained with a dye called eosin. Eosin dye to red or orange color vastundi. Ikkada some, I think, purple color with methylene blue dyes. Okay, you can write all these properties separately. About the, you have to write about the nucleus, function, lifespan. Okay, if at all you can remember, you can write the count also. Okay, and agranulocytes are divided into monocytes and lymphocytes. Monocytes are the largest leukocyte. Here I have written M. Monocytes are the largest leukocytes, and they also form the first line of defense. Neutrophils and monocytes combinedly provide the first line of defense against infections, and their lifespan is 2 to 5 days. Here the nucleus is oval or horseshoe shaped. Okay, kidney or bean shaped, anything. Oval, horseshoe, kidney or bean shaped nucleus. Monocytes, lifespan very short, 2 to 5 days. They form the first line of defense against infections. Next, lymphocytes, very short lifespan, half to 1 day, and they are again divided into T lymphocytes and B lymphocytes. T lymphocytes provide the cellular immunity, and B lymphocytes, they provide humoral immunity. Okay, and they are short-lived, long-lived lymphocytes are also there. And these two are the actual main groups. T lymphocytes and B lymphocytes, they are concerned with the antibody production or the immunoglobulins. And here this is the nucleus, which occupies most of the space, and very less cytoplasm is present, no granules. Okay, this is about the leukocytes. And finally, platelets. Platelets is the last components. These almost 2 to 5 lakhs per cubic mm. See, we heard that we are hearing during infections platelet count padipoyindi antunaru. Any infection, recently awareness has been increased about these platelets. They fight with the infection and they get decreased in number. But the platelets, if the platelets decrease, the emergency situation is created. So they are increasing, or they are giving the patient this plasma and the platelets. So they play a very important role in blood clotting. Okay, they are rhombic in size. They are cytoplasmic fragments. They are cytoplasmic fragment-like structures. No nucleus, but contain variety of enzymes. Okay, and here they contain excellent amount of phospholipids and proteins. They play an important role in blood clotting. Okay, rhombic or cytoplasmic fragments, fragment-like structures, and with this, the actual composition of blood is completed. I'll send you some PPT or some links about this. Not link, I mean YouTube link regarding. I don't think you can. You need much information about this. Just to remember, I will give you the material also. Much about remembering all the points, and you can score a good marks by writing this answer. I think all of you are aware of all these points. Just refreshing, that's all. Okay, write neat diagrams with colors, and you can get good marks. Diagrams of eosinophils, neutrophils, basophils, RBC, highlight the RBC erythropoiesis and what you call the shape and properties, and that's enough. Okay, highlighting those points, you can get good marks for this. Next class we'll see blood clotting. Okay, thank you.